So it gives me great pleasure to introduce this year's Hill lecturer, Austin Roorda, PhD from the University of California, Berkeley. He received his PhD from the University of Waterloo in 1996 with joint degrees in both vision science and physics. And since that time, he is very well known for pioneering applications of adaptive optics and ophthalmoscopy. So I know most of you, all of you, certainly know his name and research reputation. He's mapped the human trichromatic cone mosaic while he was a postdoc at Rochester, designing and building the first adaptive optics scanning laser ophthalmoscope at the University of Houston, and tracking and targeting light delivery to individual cones in the human eye at Berkeley. And he's part of the first team. See, I knew if I just kept introducing long enough, if I go through your entire CV, every seat will be taken before one. He's also part of the first team to use adaptive optics imaging to monitor efficacy of a treatment to slow retinal degeneration. He's been at Berkeley since 2005, where he's a member of the Vision Science, Bioengineering, and Neuroscience graduate programs, and heads the program in Vision Science. He's a fellow of the Optical Society of America, ARVO, and the American Academy of Optometry, and his award list is distinguished by having received the Distinguished Alumni Award from the University of Waterloo School of Optometry and the Glenn A. Fry Award from the American Academy of Optometry Foundation. So please give a warm Buckeye welcome to Dr. Austin.
All right, thanks. It's on? Okay, it's on. All right, thanks. And it's a real thrill to be here, finally. I've never been to Ohio State University. I'll show you on my next slide, my circuitous route to get here. It's an honor to have a lecture named after Dean Hill and his wife, Lee. I see Lenora, but Lee and Lenora. Supposed to go by Lee, Dick and Lee, or Richard and Lenora. Discovered the strong connection between Dean Hill and Berkeley. And in fact, I occupied a lab that where Dean Hill did a lot of his work, which was Horace Barlow's old lab. But, they always remind me of that, and I don't really want to be reminded of the history of the lab because there's a lot of pressure. But I'm doing my best. And so the title of the talk is Hacking the Human Visual System. It'll become apparent why that title is, I'm using that title. And because I'm in a school of optometry, and because there are a lot of, I presume, optometrists or clinically oriented people here, I'm going to focus on some of the clinical effort that we do in the lab. So it's going to be a little mix of clinical and basic science, so hopefully something for everyone. So I just want to start, though, with a world map. And there's, that's Columbus, I think, according to Google Maps. I grew up in Clinton, Ontario. I think one person knows where Clinton, Ontario is, Andy. I went to school at Waterloo, did my undergraduate and my PhD degree at Waterloo. I went to Rochester, New York, and then scooted down to Houston, Texas, where I spent my first years as a faculty until 2005. And then I've been at Berkeley since 2005. So I'm doing this expanding spark. And now I live in Oxford, in UK, doing a 6-month sabbatical. So that's the time that I've been invited to come to visit. So I flew here, and if I fall asleep before the end of my talk, it's not my talk, it's my jet lag. So anyway, so here's what motivates our research. This is one of the things. This slide sort of encapsulates some of the phenomena that motivate our research. And so, and I'll tell you more about the data and how we acquire these images, because these are, oh, I'm going to turn on my laser pointer, I apologize. Here we go. So you'll see the pointer. I'll use my screen, but you see it now, right? Okay, so on the left side is a map of the trichromatic cone mosaic. And this is just one example for one individual. And the sensors, or the photoreceptors, are sensitive to different parts of the visual spectrum, reddish, greenish, and bluish parts, colored red, green, and blue, just for illustration. And based on the activity in those 3 cone types, distributed in the manner that they are, which is largely random, humans have this exquisite ability to appreciate over 1 million different hues. And so we can take what looks like pretty sparse information from the sensor and generate these rich color percepts that we have. And how do we do it? And then at the same time, the role of our visual system is to process spatial information, recognize faces, read letters. And this is an example of a movie. This is a real movie of a human eye looking at a 2018 letter. And over the time it's taken for the person to tell you what the letter is, and this is a person fixating and looking at that letter. Over the time it's taken for them to decide what the letter is, or in this case, what the orientation of the letter is. The letter has moved over many, many cones and moved many times the size of the letter itself. And despite that motion, we do really well on this task, remarkably well. And despite that motion, we don't perceive that motion. And of course, everyone is aware of this, but when you do the research that I do, when we started imaging the retina on the scale that we do, you're faced with these questions head on. And you can't help but just wonder about how the whole thing works when you look at the retina on this scale. And so a lot of the work we do is exemplified here, but I'll tell you about, is motivated to sort of ask these questions. And then as a clinician, you might see something like this. This is a condition called, I'm not a real doctor, by the way, so it's called acute bilateral foveolitis, which I understand is what you call something if you don't know what it is either. And this is a patient who presents with poor vision, and then you look at their fovea, and with an adaptive optics image, you see this lesion. I'll focus a little bit more on this on later slides. And you look at an OCT cross-section. This is right smack dab in the middle of the fovea. You see this big disruption in the layers that are consistent with the photoreceptors. And so you wonder, what are the visual consequences of something like this? So it's not just imaging. In fact, the imaging that we do is what generates more questions than answers about how vision works. And so now I'll go through the technology that allows us to ask questions or address the questions that we have about vision in the way that we do. So how many of you are optometrists? Put up your hand. Okay, that's just about everyone. All right, so this is an optometry lesson. And you may have learned this if you were paying attention in your first year optics, but you probably did something about physical optics. So there's a basic property of light that tells you that as the cone of light that's focusing to a point gets steeper, the spot that you can focus to gets smaller. And it's just the law of diffraction, basic property of optics. And so in microscopy, this is referred to as numerical aperture. If you increase the numerical aperture, you increase the steepness of that cone, you get better resolution. And in the human eye, the same thing happens. If you increase the size of the pupil, you increase the steepness of the cone that's focusing an image of a point or some distant object onto the retina. And the laws of physics tell you that that focus should get sharper and sharper as the aperture gets bigger and bigger. And if you're a microscopist, just to put this in perspective, a 7mm pupil in a human eye is like having a microscope objective with a numerical aperture of about 0.2. So it's not great, but it's not bad. And the scale of the focused spot that you can achieve through a 7mm pupil in a human eye is shown here. And the scale bar, you can read it, it's kind of small on this projector, but the scale bar here is 10 microns across, or about 2 arc minutes across. And you can see that there's a pretty significant difference between the size of the blur that you would get through a 1mm pupil down to a 7. Importantly though, the focus spot that you can get through a 7mm pupil is small. It's on the order of the size of a single cell. So that tells you that with the numerical aperture that the eye offers through a 7mm pupil, you have the capacity to focus light as small or smaller than a single cell on the scale of microns, which means that if you do ophthalmoscopy or you deliver images to the retina, you can either target single cells or image single cells. And so if you only went to this lecture, You'd say, okay, I've had enough with optometry, I'm going to get into imaging, and I want to image single cells to ask these questions that I gave you in the first slide. And so you get patients into your clinic, and you just dilate them, and you try to take an image of the retina through the largest people possible, taking advantage of this simple rule. But this is what a real human eye looks like. A real human eye is not perfect at all. It's fraught with all kinds of imperfections, irregularities in the surface of the cornea, misalignments between the components, relative tilts that conspire to make the image blurrier as the pupil gets larger rather than smaller. So it has the exact opposite effect. So if you're trying to image the retina or optimize a human's vision, a typical human's vision, you're trapped between blur caused by diffraction through a tiny pupil and blur caused by aberrations through a large pupil. And sweet spot is between 2 and 4. Not so bad. The point spread function is a little larger than a single cell, so it's actually really good and almost optimal for human vision, but not optimal for a scientist who wants to look into your eye. So this is how adaptive optics works. I'm going to tell you in one slide. You measure the imperfections that give rise to the blur. So we do that with wavefront sensors. You've probably heard about them, or you may use them in your research, even in your practice these days. So you measure the imperfections, and then you develop an optical component, or you employ an optical component that has equal and opposite imperfections. And you take the light that comes out of the eye, you pass it through that component, whatever type of component it is. They all achieve the same purpose, and they cancel each other out. Just like if you're myopic, you have excess of power in your optics, you put a lens with an equal and opposite magnitude of the refractive error, and it corrects. So that's adaptive optics. And what it does is we measure over the largest people we can, we compensate for those imperfections, correct for those imperfections, and then although a natural human eye does not give us the kind of access that a diffraction limited eye does, when you use adaptive optics you can. So, and the first group that did this successfully was in Rochester in 1996. So that was part of my circuitous route, and I was the luckiest postdoc in the world, I think, in vision science at least, because I showed up just when Don Miller and Jinjong Liang and David, of course, who led the lab, finished building the system. So I got to use it. And everything I produced was gold. People loved it. They would file into the talks to see it, and they gave me way more credit than I deserve for doing anything technical with that system. But I did get engaged in basic science in that lab. Here's the one of the important slides, and this is an old slide. This is all the way back from 1996. But it makes the point clear. The contrast is a little funny, but I think it's okay. So if you take a good image, carefully correct defocus and astigmatism, and take a high magnification image of a small patch of retina, this patch is about 150 microns across, or about a half a degree of field of view. You can see some structure under cases where you carefully correct them. But when you turn on adaptive optics, it reveals the structure very clearly on a cellular scale. And the important thing about adaptive optics is it does not just make the images sharper. It's a paradigm shift in how you use ophthalmoscopy to study the eye in health and disease. Because it turns it from a ophthalmoscope into a microscope. And as soon as you can see single cells and resolve single cells, you can measure their properties, measure their function. And it just changes the way you approach, changes the way you tackle the questions you have about vision. It sort of puts it on the scale of a physiologist, except that you can do this in a human eye. Okay, so now I'm going to step a little bit back again. And I always admire optometrists and ophthalmologists' ability to look at images like this and diagnose a disease. So at the risk of, that's not a very good projection, is it? Is there anything funny about that image? Anything interesting? Can you identify what disease this patient might have? A little what?
There's got a little schmutz in the middle.
That's just on the camera or the slide.
She's not an optometrist.
Schmutz. And have words to describe everything too.
But no one else knows or you notice.
There's a little loss of transparency here. Do you notice that? I know, I don't, but I've been informed about that. And then in a fluorescein angiogram, you can see a little bit of leakage, right? Diffuse, that's obvious. Any idea what the disease is? does the temporal retina feel? Temporal retina, sorry, temporal to the, no, nobody? What? You're nodding like you know.
No, I'm agreeing with him, and I wasn't going to say that.
What did you say?
JXT,
I don't even know what that is, but.
juxtafocal telangiectasia.
Juxtaphobia.
Yeah, I think so. I think it's called macular telangiectasia.
Yeah.
Very good. All right, good, good. So anyway, this is a disease that we've been funded to study, and but I'm going to zoom in a little closer. So we've studied patients with this disease with adaptive optics. So we're going to zoom in here, and then zoom in. So this is a zoomed in version of a fluorescein angiogram, and as you've probably seen from Stacy and Nathan's work, we can take adaptive optics images and assemble montages to generate reasonably wide field images but of microscopic structure in the retina. And so this is a close-up of this patient's retina. And this is their fovea. We can identify their fovea through some functional tests, but this is a scatter plot. These are just white digital marks that indicate what part of the retina they fixate with. This is a lesion here. And then you see some other things like little microcystic structures, and some of the abnormalities in the vasculature sort of appear here. But there's all kinds of rich information in this image. And then we often do OCT in conjunction with our adaptive optics. This is just conventional OCT. And you'll see this is where we do the OCT B section is coming from. And you'll see that this lesion that I identified here appears clearly in the OCT as a total break in the inner and outer segment junction layer. So by all accounts, this is a part of the retina that is devoid of cones, and a scotoma, or a blind spot. Now, we had only done structural imaging of this patient at the time. And, but the question that I have is when I see this, okay, what is their vision? How can I quantify? I can see a lesion, but how do I quantify their vision? And if you're an optics nerd like me, then you look, and we love imaging, and we love looking at the images, then you look in a little closer, and then I want to ask questions about this cone. And what is it about this cone that makes it reflect so nicely, and about whatever's happening beside it that looks completely dark, right? So we want to probe and study vision on this scale. So this is a platform that we use. It's the Adaptive Optics Scanning Laser Ophthalmoscope. It's just a scanning laser ophthalmoscope with adaptive optics to improve the sharpness and contrast of the image. And for those who aren't familiar with a scanning laser system, what you do is you form an image in a scanning laser system by focusing a spot on the retina, measuring the amount of light that comes back from that spot, and then moving the spot. So you build up an image pixel by pixel as the beam is scanning across the retina. So you build up a frame, an image of the retina over time. And then we do it continually. The scanners are always happening, so we can record a video of the retina. And there's lots of features about scanning laser imaging that make it really useful for the kind of research that we do, which you'll appreciate as we go through. So the next 4 slides are going to show about 15 years of work and how we went from a basic platform, which was scanning laser ophthalmoscopy, to a platform where we could study and ask that question about that cone, and does it still work? So this is adaptive optics in action in an SLO. And so when you turn on the adaptive optics, the image gets sharper, it gets brighter, and it gets more contrasting. All of those things happen in the image. And if you're focused on the right layer, like this one, you can see the photoreceptors. So all the small spots are light reflected from individual photoreceptors. This is an image 360 microns across. It's about 1 degree away from the center of the fovea. So that's adaptive optics in a scanning laser system. The other thing you'll see is the eye is constantly moving. Even though you have an eye, ask the patient to hold still. The eye is never still, it's always in motion. And you'll see some blood flow moving through. The motion that you see are red and white blood cells and the gaps between them going through the microvasculature. So I won't focus on that, but what I'm going to focus on instead is an ability of a scanning, some feature of a scanning imaging system that make it especially useful for the kind of research that we do. So as you're scanning, imagine now, as you're scanning, you can control the scanning, the intensity of the scanning beam. So while it's scanning, you turn it off at appropriate time points to put an image onto the retina. Sort of the same way you put an image onto a CRT screen or old television sets. You modulate the scanning beam. So So in this case, let's say we're imaging with red light. So the patient would see a red square that covers the area that we're scanning. And if I turn off the laser at the appropriate time points to render a letter E, then the patient will see a red square with a black letter E within it, right? I'm just putting that image directly onto the retina. But now if I turn off the laser that's doing the imaging, that pixel doesn't record any light. So I get no intensity, so the image that I deliver gets embedded or encoded directly into the video that I've recorded. So the letter E that you see on the video is not an overlay of an impression of what I'm doing, it's the actual video that I record, because the laser's not on during the time points that I'm delivering the stimulus. And so I get a recording of the event. So now I'm going to be delivering doing a visual acuity task. I'm going to put a letter on the retina, I can make it progressively smaller, ask them what the orientation is, which is the way we generally do our visual acuity tests. And so I can record their visual acuity. But the nice part is I can now see exactly, for each task, I can record a video and see exactly what photoreceptors have interacted with that stimulus, exactly how the eye has moved over the time course of presenting that stimulus. So you can appreciate that I have a really nice, powerful tool for relating structure to function. But the eye is always in motion, and if you want to target that cone in that patient, we need to be able to track the retina. And so we've, again, developed tools with a lot of help from my colleagues at University of Houston and Montana State University to stabilize the image that you're recording. And if you stabilize an image that you're recording, the byproduct of stabilizing it is knowing how the eye is moving. So we can generate a stable video, and that's illustrated here. And by stabilizing the video, you're basically removing the whole frame of it. It's even more nuanced than that because of the scanning, but I won't go into detail about that. But the byproduct of stabilizing the video is this really accurate eye motion trace. It's accurate in the sense that it's accurate on the scale of a single cell. So we can track eye motion to within a fraction of a micron. Okay, so that means we can track. But now we have to do it really quickly so that we can time and control the stimulus to land on a target. So we do that by combining the stimulus delivery property with the stabilization and tracking. Now we can control the timing of when. So as you're collecting an image, you're estimating looking at where the retina is, and then you can control the timing of the laser that's scanning to deliver an image at a targeted location. So now this is again a video, it's not a rendering. It's the actual video that we record when we're stabilizing a letter E on the retina. And you'll see it's locked on pretty well. If you look carefully, it's not perfect. It's wiggling a little bit because you don't always get a perfect estimate of where the eye is. And there's some latency between your last estimate of where the eye is and the eye might have moved in that time. So you can't do it all instantaneously. But the wiggle is small because we keep the latency down. And so the standard deviation of that wiggle is a little smaller than the size of a cone. So we can track on that scale. So now we have a tool to deliver stimuli in a live human at targets on the scale of a single cell. And again, this is a case where we're imaging with visible light, and then we're turning off the scanning laser to put it'd be like a black letter E on a red background. But of course, there's a lot of things you want to do differently. So we've built systems that combine multiple wavelengths. So now I could be imaging in the infrared, which would be relatively dim, ideally invisible to the patient, so they wouldn't know that they were being imaged or tracked. And then at the targeted location, once I identify the cone that I want to target or whatever I want to target, we turn on a green laser instead. So the subject would see a dim or no field that's doing the scanning, but they might just see a green spot landing on the target. And then this is persisting at the same location. What do you think happens if I keep a stimulus locked on a target, stabilized? Disappears. Yeah, in fact, we know it's working when you don't see it. As a subject, you know it's working when you don't see it. But it disappears if you leave it for any long period of time. But initially you see it. And so there's lots of experiments that we can do with stabilized stimulus delivery. And I'll show you one example here. In fact, this was the first sort of technical demonstration that we can do what I just told you we're doing. And this is where we target cones in the mosaic. In fact, here we're targeting a specific cone with green light, and then we're targeting a space between the cones, which we call a gap. And we can deliver light to that cone, ask the subject what they see, and then find the intensity level for which they can only see it 50% of the time. In other words, we're finding the sensitivity threshold for that specific targeted location. The idea being that if we deliver a light, if it's small enough, if the spot is small enough, then you can focus light onto an individual cone, and if you miss the cone, they ought to be less sensitive to it than if you hit the cone. And so that's shown, and it's not perfect because not all the light gets into a cone. There's still diffraction, so light does spread around. And if you miss the cone, that means that you're getting light into a bunch of neighboring cones. But what happens is if you present the light to the cone, here we're sort of hunting around for the light level that they can see 50% of the time. And if you put the light on the cone, it takes less light for them to see it 50% of the time, so the threshold is lower than if you put the light between the cones. And the amount of change here is consistent with an optical model that suggests that our focus spot is really, really small. So we can do it. We can see cells, track cells, target cells, and do visual psychophysics on individual cells. So this is what we call hacking the visual system. We're putting things on the human retina that the normal optics, the normal eye cannot. We can control for the optics with adaptive optics. So we can focus the beam smaller than humans evolved to do. We're focusing a spot smaller than humans need for the spatial vision that they have. We're controlling the motion. So the eye naturally benefits from its own motion, but we're removing that so we can target individual cells. So in that way, we can test the retina. So we've sort of hacked the visual system. We've done things that normal optics and normal human cannot do. And so it gives us a new avenue for testing certain properties about human spatial and color vision. I don't know how much time I have, but I feel like I'm going slow. I'm a half hour in. Okay, I think I'm doing okay. I'm doing all right. And I can stop any time. It's like short stories. Every story ends and the new one is brand new. So if I get stuck. But feel free, if you have any questions or comments, feel free to interrupt and put up your hand and just yell or do anything anytime. Okay? Because the more I hear from you, the more I know I'm getting through or not getting through. Okay. So I'm going to talk about a couple of projects on basic science. And I'm going to focus first on color vision. And I have just so blessed to have such a great group of people that work with me. And I can't do any of this stuff. And that is the absolute truth. And in fact, nobody in the lab can do everything. I'm a little bit good at everything. But everybody I hire or works with me is better at me than something. And that's the way you build a lab. Always hire someone that's a better programmer, or a better clinician, or a better psychophysicist, or a better optical engineer. And if you do that, you'll succeed. And my job is just to pick the people to hire and direct what they do. And so I've always had a really great team. And in fact, one of the guys you'll recognize, Will Tooten, he says hi to everybody, by the way, so I take this opportunity to. And I'm thrilled that he's come back to Berkeley. Your loss, our gain. But I told Carla that we're nurturing Will to be the dean. So when it comes time for you to retire, then he'll take over. Right? Or he'll be a candidate anyway. But he's great. And as are everyone else here in the lab. And so I'll show pictures of people involved in these projects as I go through, and I'll promise to get back to it in the end. So to study color vision, years ago we had mapped the cone mosaic. And mapping the cone mosaic would help launch my academic career. But it led to way more questions than answers. It didn't tell us anything about color vision. It just made us confused about color vision. And it was like right up there up front. That's the cone mosaic. How do we see as well as we do? And so in order to study color vision, we had to learn again how to map the cone mosaic, which Ram Sabason, who is a postdoc in my lab, did. And so S1 and S2 are actually two postdocs in my lab. So we need to classify their cone mosaics so we can study properties of their color vision. So we'll still call them S1 and S2. S1, though, happens to be initials WT. So allowed to tell you that. It's personal health information, but I think I'm allowed to tell you that. Anyway, that's Will's random. So here's one of the subjects. And so we classified the cone mosaic, but of course we wanted to ask more. And so now I'm going to just zoom in on this, and we have a cartoon that illustrates one of the first experiments that we did related to color vision. And so this is the cartoon. And so we could target, so we knew the cone mosaic. Of course, you don't see it colored, but we had maps, so we could figure out where they were, and that takes a lot of careful approach to do it. But these guys did it. So then we could target the cones, and in this particular experiment, when you flashlight onto a cone, the subject sees the spot, and they can actually see the color. And so we forced them to tell us what they saw. And in this particular experiment, we gave them 6 choices: red, green, blue, yellow, white, or I didn't see it. And then every time we flashlight onto a cone, they would report their response. And then we'd hit another one at some random location. And then we hit a whole collection of cones as many times as we could to sort of collect responses, multiple responses from each cone about what they saw. Okay? And what you see here in the movie are just some example. They might actually be real, but either way, they sort of illustrate the point at the start again.
Did S1 report scarlet?
We didn't let them do. It doesn't want to play, but when they played, they would report. If you hit this cone, they might say green, or they might say white, or whatever. Anyway, this plot shows one of the results from this, and it shows the cone mosaic, and then around each cone there's a pie. It's a histogram of the responses. And so, interestingly then, if you look at some of these, so this cone, every single time we hit this M cone, a greenish sensitive cone, 20 out of 20 times the subject said green. And this one right beside it, 19 or 18 out of 20 times they said white when they were forced to make that judgment. And here's one, an L cone. This one is a strong color reporting cone. But the one right beside it, white every single time. So these are extremely robust, repeatable responses. And when you're a subject in the experiment, like Will says, I thought I was just making stuff up. Like I'm forced to come up with 1 of 6 responses for what I see. And although he feels like he's making stuff up, you plot it, and it's remarkably robust. And so a few observations, and I won't go into detail because we don't know what to say about this. It's, again, everything we do leads to more questions than answers, unfortunately. But the light was always green because the laser that we used to stimulate was 543. We chose 543 because it's equally sensed by the L and the M cones. It's right at the intersection between their spectral sensitivity curves. And so, but that's a decidedly green color. You look at it and say it's green. And so the flash itself, the light was always green. When you stimulated L cones, or the red sensitive cones, they mediated, almost invariably mediated sensations of red if they mediated a sensation of a color at all. So you'll see all the red cones, with few exceptions, but all the L cones are red sensing, and the greens elicit a sensation of green. Stimulated M cones mediate sensations of green. Some cones are strong color reporters, though, and others are not. And I guess one take-home message from this is that the retina has a dual role of encoding space and color. And if you look at the evolutionary time scale of the development of human vision, the subsystems that subserve spatial vision, center, surround, and all that, center, surround of receptive fields, midget and parasol receptive fields. It turns out that if you just add different cone types into that system, it will provide information about color to the brain. And so the color system seems to ride, in part, on top of an existing system that subserves spatial vision. And so maybe it's not surprising that the brain then will attribute some cones as strong color reporters, but most cones, it's not doing anything but just encoding spatial information. But again, we don't know.
Couple questions over here.
Yes.
Liz.
Yeah, go
Sorry,
ahead.
how would you explain the cones, like the one down on the bottom, the M cone that is exhibiting white and red? So it's the subjects were reporting.
Yeah,
They weren't reporting green. They were
so
reporting something
that's right. So we don't know,
different. Okay.
but we don't put a lot of stock. We're looking at because it's such a new area, we don't know whether that's a misclassified cone or some noisy data. So we're focusing on the main trends. Not to say that those are not important, because it's very possible that that is a cone that indeed elicits sensation of red. But we haven't gone further to study that carefully. But it's a good question, Tom.
Yeah, for those cones that were not strong color reporters, like, did they tend to be surrounded by cones of the other type, and there might be a little bit of leakage to those, and so the composite response might not be?
Yeah, that's there is a tendency for cones to be in a mixed surround, although you think those would be the cones that would be wired to be specifically useful for color. There is a tendency for those to actually produce more achromatic responses. But we've done careful optical modeling. We can't prove it, but we have every reason to believe from the optics that it's not due to leakage. And we've modeled the blur that we have and how much information that would be sending to the neighboring cones. And so we don't think that's the case. But it's not entirely out of the question. Anyway, this is but food for thought for us. We puzzled and scratched our head about this. And these are really tough experiments. We're tracking cones, and as a subject in there, your ability to actually respond to these like stimuli, which are just really brief flashes that are almost invisible, is hard. And so you only get an hour out of a subject before they just start freaking out. They just don't want to do it anymore. And so, but it took hours and hours and hours of sitting in the bike barn, which Will is always very patient. He's happy to do it. It begs the question then about, there's like 100 questions you ask and 100 new approaches that one might take to further study these phenomena and take it to the next level. And Brian, who is a postdoc in my lab, he's written a couple of papers. He did more sophisticated color reporting, so he would report hue and saturation on graded scales. And so this is a nice paper that describes that. And then we studied sensation from M cones, and they're relevant, how they're spatially related to S cones. I won't go into detail on that. And then he started looking at how pairs of cones combine to elicit a color experience. So if you stimulated L cone beside an M cone at once, what is the color that you see? And so these are some follow-up work that we had done. But it was daunting to think about how we're not even sure. We're puzzled about how to tackle or what questions to address and how to do them, and they're very time-consuming. And so, but we're working on it, and as is shown here by Brian's papers. But I'm going to step back a little bit, and this is where sometimes someone else can get you out of the little rabbit hole that you've crawled down, and you're focusing on, like, what are you doing? You're focusing on what 1 and 2 cones do. This is not studying human vision. This is just following your little obsession about what cones do. And Ren is an engineer at Berkeley, and we've thought about this, but he articulated this whole thing to us. And he's totally thrilled about our capabilities. That I give him credit for motivating the work that I'm going to describe now. And this one's kind of fun. So I'm going to take you through a thought experiment first. And then I'll show you some experimental data that supports this. But you have to think about how color vision is, how percepts of color are generated. And so you're looking at a red square, and it looks red to you, right? It looks like a uniform red square. And that square is landing on a patch of your retina that's comprised of 3 cone types. And if I know the wavelength of the red square that you're looking at, and I know the spectral sensitivity of the cone types, which we know very well, aside from slight shifts in their peak sensitivities, we know that. And I can take a 600 nanometer light and figure out the exact excitations of the M, L, and S cones of that 600 nanometer square that's landing on it. So I can just generate a grayscale map here of the same photoreceptors showing their relative excitations. And you'll see the L cones, which are red sensitive, are brighter because they're more activated by that light than the green, than the M cones. And then the S cones are almost untouched. Now, when the brain is faced with this input, when the brain gets this sensory information, it attributes it a percept of, okay, do you see a luminance variation? It's a graded variation of activity across the retina. So you might see a luminance variation, maybe slightly blurred because you don't see your cone. But we don't do that. No, we immediately and effortlessly convert that signal that comes from the sensors into a perception of red. And nobody sees anything different than that. Now, your red may be different than mine. You can argue about that forever. That's irrelevant. Everyone paints that sensory input with a uniform attribution of color, and that we call red. Okay, so, all right, that we know. Now, I'm going to take 525 nanometer light, and I'm going to look at all the cones in the mosaic, and I'm going to modulate the power of I'm going to deliver microdoses of light to every single cone in a manner that's consistent with their type. And I can figure that out. I know the cone spectral sensitivities for that specific 525 nanometer wavelength. And so I can deliver microdoses of light to every cone to generate identical photoreceptor outputs, right? That's just math. So I can generate an identical photoreceptor output to send to the brain. So what are you going to see? Are you going to see a patterned green model-y light distribution? There's no way. You're going to see a uniform colored square. If you don't see it, that means we're not doing it right. Because all the brain has access to is the signals from the photoreceptors. It uses no more information than that to generate this perceptive color. So what we call this is spatial metamers for color vision. It's a metamer. It's something that gives you an equivalent response by generating the same sensory signals. So we're totally excited about this. It pushes the technical capabilities of our system because now you have to deliver over multiple cones. But we've done it on a reasonable scale. And so this is a patch of one of my postdocs' retina, not Will this time. And we need the cone mosaic. And now we're going to take 543 nanometer light, and I'm going to deliver this pattern, where the hot spots lie directly on top of the M cones, the green sensitive cones. So I'm going to generate sensory inputs from that light that are consistent with the color green. And the subject sees, it's a model-y pattern, because we're just targeting the M cones, but the subject reports a fairly uniform field that is green colored. Maybe not that surprising because it's a green light to start with, right? It's 543. Okay. Now I'm going to do the L cone weighted. So now we take the L cones and we pattern a stimulus with 543 nanometer light, which is decidedly green. Flash that onto the retina, and he reports a uniform patch of red. So we're heading in the right direction. We can predict exactly what colors they should see because it's just math. We know the spectral sensitivities. We know what the excitations are for any color on the L, M, and S cones. And what we try to do is generate a range of sensations across the whole visual space. But what I'm showing here is that just by tailoring microdoses of light to the specific cones in the mosaic, I can give this subject sensations all the way across this part of the spectrum from reddish to greenish. And I think the only reason we're not going further is technical. Some of it's technical. There's also some other fundamental bits of information that the human gets that helps inform us about color. I'll touch on that a little bit later.
If you do the math wrong, does the color change or does the uniformity fall apart?
Here's what, yeah, so uniformity is key for us to because uniformity is one thing, one attribution that you give given those sensory inputs. So two important aspects of what the subject saw was uniformity, which we haven't been able to quantify psychophysically. It's just qualitative description. But he did a color match to get the color that we saw, and color. And if we scramble the stimuli, so if you scramble them, you just stimulate random cones. He should also not see color. It should not appear green because it's a scrambled input, which is not consistent with green. He should see a luminance, very speckly luminance variation. And what he saw, if we scrambled the stimuli, then he saw a motley pattern that was kind of achromatic but kind of greenish, in a way that this was not quite the color that we wanted to get, but we did spread it out a little bit. So we're getting close. Yeah.
Having spent a great deal of my youth hanging off a bite bar too,
Yeah.
doing two-color intimate threshold experiment under the paradigm of overstanding styles, I can tell you that when you view a flash of light under M-cone isolation conditions, that light looks blue, not green. And also, in agreement with this, the color appearance to the tritinope, which has actually been studied using a unilateral, very well characterized unilateral tritinope. The appearance of green lights is blue. So I will give you, this isn't a challenge to your results, of course, but I think that there will be some very interesting science involved in bringing together the various sources of data on the appearance of colors.
Yeah. No, I agree. Yes. Yes, that's. I'm just thinking of one thing. Brian Schmidt did some predictions of color under M-cone isolating conditions, and indeed it does shift in the. It's predicted to shift in the blue direction. I'm not sure to the extent of what you're describing. Right. But there's a lot of mysteries about
There's a
this.
great deal of mystery.
Okay. So again, do we have all the information required to generate colors and percepts just by hacking the system in the way that we do? Have we, oh, here we go. Have we really hacked the visual system? We have hacked it, but can I hack it in a way to generate any color experience? Have I done it suitably? And have I thought of all the things that one needs to think about? Have we fully reproduced the sensory signals that inform the brain about color? And I told you that I can, if I know the cone mosaic exactly and I can deliver the microdoses exactly, you might think yes. But there's one important component missing, and that is motion. And in fact, the moving eye, when a trichromatic mosaic moves across an object, it gets new sensory information that helps to decouple luminance information from color. And I don't have the time to go into all the details, but I'll show you a slide a little later that might help you appreciate that point. But there's certain signals that might be misconstrued as luminance that sort of travel with the retina differently than a signal that's consistent with a color. Anyway, so but I can just talk specifically about the benefits of eye motion for spatial vision. And this is done with this crew of people. And this asks the simple question. And the simple question is, are fixational eye movements beneficial for vision? And I showed you that I can put a letter E on the retina, just fix it within the world. In other words, just write it to a very specific location of the raster. And it would look just like a letter acuity chart, which it does. Or I can stabilize it on the retina. And so under what conditions is their acuity better? Wouldn't it be better if they just held their eyes still? And so wouldn't it be better if I could stabilize the image on the retina? Now, the problem with stabilization is that it fades from view. So we have to control for that, which we do, by presenting it briefly enough. And then also, even if you present it briefly, a stabilized image has lower contrast because it immediately goes down in contrast from the moment you present it. So we can present the natural stimuli at slightly lower contrast to balance it against that. And when you do that, you measure acuity. It turns out that this shows 4 subjects, 1, 2, 3, and 4. And what we do is an acuity test. It's a tumbling E test, and we make it really challenging. We make it so that they're just above or about 50% of getting it correct. Chance is 25% on a 4 alternative force choice. And if they get 50%, they're just marginally above chance. In fact, they're all a little lower than 50% here, but above chance. And the purple bars show for a fixed letter size, so we set the letter size so it's about 40% correct. And then we compare how they do if it's stabilized versus if it's naturally moving. And it turns out that across the board, every subject did better when it was moving across the retina than if it were stabilized on the retina. This is not surprising from an image processing point of view, but the fact that the human has the neural machinery to synthesize that information over time as it's coming in to achieve this on a very fine scale, on a photoreceptor scale, is it's interesting to us, but it's just an interesting aspect of human vision. And if you think about the letter, and this is an actual representation of the letters that we're presenting. Look, they're close to the they're close to what the sampling limit might be. You can see there's just a handful of photoreceptors picking up signals from that letter. Right? You can see the exact ones that are picking it up. And so we can model, okay, well, if it was static, then you just have a handful of photoreceptors, and these are Voronoi plots which show the gradation of excitation of the photoreceptor. So you put a small letter E on a handful of photoreceptors. Some respond like strongly because a lot of the E lands on it, and some respond weakly. But these are 4 different E's sampled by a sparse photoreceptor mosaic. There are 4 different orientations. So can you tell which one is up, down, left, or right? If you guessed, we might be slightly above chance if we guessed, right? Now I'm going to show information over time. So what I'm going to do is move the sample behind the letter. Okay. Each one of those snapshots is just as bad as the static, but collectively you can integrate and synthesize that information over time, which humans are really good at doing. And the E is quite apparent. I hope it's quite apparent. So this is how the human visual system really takes advantage of this temporal information to process for perception. And it's a dynamic process. And I always say it's analogous to, it's maybe not surprising that you slide the sensor across the image to get more information. And we do this all the time. If I see someone's nice jacket, I touch it and say, what is that, mohair? No, you don't just touch it. You slide your finger across it, right? You say, oh, is that mohair? Yeah, no, it's not mohair. It's artificial, but this is an example of dynamic sensory information that reveals way more, has the potential to reveal way more than static sensory information. And the human clearly has the machinery that's capable of leveraging that. And so fixational eye motions, we've shown, we're not the first to show this, of course, but we can show it on a scale that is quite unique, and in a way, in a manner that's quite unique.
There might be an analogous thing with Braille readers. I don't know this for sure, but I think a little
Oh,
movement.
I have no doubt, yes, that if you just put their finger on them,
Yeah.
it would just not.
Yeah.
Yeah, in fact, that's a really good example, because they're actually resolving. Here I just get statistics, right, of the surface, but with Braille you're actually resolving, you need to resolve the spatial detail. So it's a good example. And of course they benefit from, there must have been a study, but I'm sure they benefit from sliding.
So which part of the retina was that? The fovea or peripheral?
This was just off the fovea. There's a couple of reasons for doing it. Technically, it's easier because we can resolve the cones better there. Practically, it's important to do it just off the fovea because when you want to test a stabilized image, if you put a stabilized image on the fovea, they tend to chase it, like you chase floaters that you're looking at. And that would totally disrupt the task. So what we do is we have them fixate on a target, and then at 1 degree away, we present the letter, either stabilized or not.
Okay.
Yeah.
And then, the early work by David Burr talks about motion smear. So what kind of velocity is this?
This is natural.
Oh, which
Natural
is about
motion.
what? I don't remember.
Pardon me?
Natural miniature eye movements. What's the
Yeah,
velocity?
I don't know. It's small, like it's, so a saccade would be 3 degrees per second. So these must be just a
Okay,
fraction
alright.
of a degree per second.
So there's no motion smear, it's just,
Yeah, but I'm not even sure what motion, but no. I shouldn't say I'm not sure what motion smear is, but it's not a scale of motion that we're
that's like
interested in.
15 degrees
We're looking
per
solely at
second.
fixation. So my point here, and going back to the color story, is that we have this remarkable ability to synthesize this dynamic information, but we have this sparse or randomly distributed trichromatic mosaic. And this is just a really simple cartoon. But processing color on a fine scale, which are experiments that are underway in my lab, may also be leveraged by motion. And in fact, if you move the trichromatic mosaic across a colored object, then you can collect the SMNL information at each point across that image to extract the color. And this just shows an illustration of a cone mosaic, trichromatic cone mosaic moving across a colored eve. At any instant in time, you don't know what the color is. But over time, you can integrate that information to extract the color. And I fully believe that temporal information is used by humans to inform us about color. It's one aspect. And so one of the reasons we may not have gotten the colors that we expected fully when I showed you what we did with that subject, giving them red and green sensations, is because that was an image that was stabilized briefly on the retina. And they didn't have the sensory information that might tell them, dissociate color from luminance information, so they may appear less saturated as a result. So we're developing tools now, luckily with good people in electrical engineering, to sort of generate more realistic stimuli, not just a patch that's fixed on the retina, but allowing the retina to move and peruse the object, but then tailoring the stimuli within, tailoring the cone, the doses within the stimulus, like the apple, appropriately for the cone mosaic that underlies it. And so we think that in this condition, sorry, it stopped playing, but so motion is the whole part. So how could it stop playing for me? That's what the PowerPoint does. There we go. So what you have to do now is track the retina, know the cones, know when the cone is lying in a specific area, and deliver the microdoses appropriately. And we think that under these conditions, then we have fully recaptured or recapitulated the sensory information that the brain has access to. And then there's no reason to think that they wouldn't see exactly what they would see if it were a naturally colored object. Okay, I have 0 time. But I want to talk about one, because I need to talk about some, I don't know, what should I do? Are people ready to go?
5 minutes good?
I'll take 5
I
minutes.
took 5 minutes to introduce you, I'm sure,
That's right.
so there you go. That's my gift to you.
So I want to go back to the clinical example, because we do spend a lot of our time doing clinical work, and largely thanks to Jackie Duncan, who's an MD at UCSF. We've deployed a system there to do a lot of the work that we do. And we were interested in this cone, because now we do sort of microperimetry on a cone scale. And so does that cone still work? How does it work? What does it look like near there? And we brought this patient. This is a patient that we imaged in 2011. And then we brought them back in 2013, once we had built the machinery to do microperimetry. Because now because I've been thinking about that cone for 2 years. I wanted to go back and test it. And we imaged the patient again. And this is the montage that we got of the patient the second time we saw them. And we're obsessive in the lab about aligning our images. So these images are perfectly aligned, and you'll see there's little microcysts here and some blood vessels here that can give you confidence that they're aligned. And so, but you get a hint about the lesion, and I'll zoom in on it, but everything changed over the course of those 2 years. They got a little bit worse, but things just changed. They just looked different. And so this is what they looked like in 2011, so I'm zoomed in. This is the part of the retina they fixated with, or the PRL, coincided with their anatomic fovea. This is the lesion here. So you can look at the fuzzy black outline. That kind of outlines an area where we don't see cones. And then this is what happened when we saw them in 2013. And so they shifted their PRL because the lesion started affecting the area that they had originally used. And then the lesion kind of expanded. Some showed up here, some showed up here, here. What's interesting is this area. Look at these bright, healthy, happy-looking cones. They were not there the first time we saw them, and they reappeared. They regained reflective properties. We're not saying they grew back. We're contending that they were never gone in the first place. They just changed whatever structural properties that would give rise to reflection. But when we did microperimetry on this, so then we selected areas and tested the vision. So we're going to measure sensitivity across the retina. And I really wanted to study MACTEL because I was convinced that they were completely blind where we didn't see the cones. I thought this is a case example of a step function. I see the functioning cone, and then I see no function. So we test at targeted locations. Here. This shows the test, and all you need to know is that red is bad and green is good. And if it's absolutely red, they have essentially a scotoma. But here, in an area that by OCT and that by our imaging looked completely devoid of cones, we saw functioning vision. Where the cones were recovered, we saw them functioning very normally. So this is a case where we don't see cones, but there is measurable function. And we don't think it's coming from anywhere else. It's not like that's leaking through and getting picked up by neighboring functioning cones. We think the cones are there, they just changed their structure. So we see measurable function in regions that appear devoid of cones. I'm going to go back to this case, which is the acute bifoveal foveolitis. Here's a case where that looks to be a lesion. You might predict that they have no vision at that location. But when we tested vision at that location, we see they function, although albeit reduced, they see function. And then we did a more sophisticated test. We tested their vision. We presented the patient with a letter. Here we go. And we did an acuity task. We do it simply. We just present them with a really challenging letter and measure how often they get it correct. And then we could restrict the trials that we analyzed to only those trials where the stimulus landed within this apparent lesion. And if it's landing within the lesion and they have no vision, then they have to be chance. But out of 100 trials that we did, and these are the traces, the red show ones that were correct, the green show ones that were not correct, they got 48% correct. So by sensitivity measures and by spatial vision measure, they have cones. The reason I wanted to get to this slide was one, is the OCT has a very specific appearance in these conditions. There are areas that lack reflection, but they're absolutely, completely transparent. The retina is not collapsed into that space. And if you see these clinically, if you see this condition clinically, it may not mean that they have a scotoma. If you see a phenotype whereby the things just stop and then you see a clear area here, those may be recoverable cones, and we've seen them spontaneously recover, like in that MACTEL patient. So this is an important state of degeneration to look at. Ultimately, these patients may lose their vision entirely, but there might be a way. I don't know how to do it, but somebody might. If we can identify cones that are present and potentially recoverable, there might be a way to spur their recovery. And it might be a common pathway in the death of a cone that could be recoverable, regardless of whether you have macular telangiectasia or some other condition. And the reason I wanted to show this was I'm going to show something that's a little off color here, not off color, but it's Will Tootin that made this slide. So it's his fault. So we see this new phenomenon, and at night we're having a meeting and we're saying, how do we describe these things? They're functional, unreflective cone receptors. So usually they come up with an acronym, and we didn't think there was a good acronym to use, so we called them dysflective. Cones that function, but they lack a normal reflective property. And I think it's an important distinction to make that we're uniquely able to look at. but I think it's a very important phenotype clinically in cases of these types and maybe many other types of degeneration. We're looking more at this. So I'll finish here. I'm going to just tell you that hopefully I made a case that adaptive optics and eye tracking, those two combined, enable measurements of structure and function in healthy and diseased eyes on an unprecedented scale. And I've shown you a bit about how our tools can shed new light on spatial and color vision. And I've shown you a bit on how they shed new light on retinal diseases, stages of retinal diseases. And I have no doubt it's complicated, but I have no doubt that tools like this will have an important role in treatment. We don't treat disease, but we can have an important role in evaluating efficacy of treatment and identifying patients that are most amenable to treatment. I'm presenting the work, but there's a huge amount of people over the years, and it's been many, many years of work and many, many taxpayer dollars that have supported it. And I showed highlights and pictures of the people, but I'll show it here. Most of our funding is from the NEI, so we're very grateful to them. But thanks for your attention, and sorry I went a little over.